record for the first part if they miss. Right, I think we can start the first lesson. Last week we have completed the mitosis. This week we can do a second type of division is called meiosis. This is also called reduction division. A reduction division. Okay, we have learned the mitosis. I think some of the information we can directly get for the meiosis, and we can learn only the important steps of meiosis, because some of the events which are very similar to mitosis. So we will try to understand only the important thing in meiosis, right? One thing is, when we learn in the stages, we learn prophase, metaphase, anaphase, telophase. In mitosis, we learn prophase, metaphase, anaphase, telophase. Meiosis, we also have prophase, metaphase, anaphase, telophase. Then it is named as prophase 1, metaphase 1, anaphase 1, telophase 1. Right, next end of this telophase, there are two cells formed. So let's see overall what is the number of chromosomes, right? So let's take a human cell. If you take a human cell, 46 chromosomes double. So 46 chromosomes double, that means it produces 92. So this cell has 92, and the 92 chromosomes basically exist in this way. Okay, now you see the one with yellow color, that's exactly the duplication of the first one, that called chromatid. This one is a duplication of this. It's a chromatid. That's why we consider as a 92. The completely red color one is the one first molecules of DNA, and it is replicated to make the second version. Then it's a copy of the first one. If this is the first one, this is the second one. First one and second one, it's a copy. Second one is a copy of the first one. So then we have a 92. If they make a 2 cell at the end, 1, 2. So what is the outcome is one get 46, other one is get 46. So this cell should have a 46, this cell should have a 46. Right, from here to here, we consider it's a reduction division. Why it is called reduction division? The chromosome number become half. Chromosome number become half, right? But if we look at the numbers, we don't see like that. We see 46 and we see 46 here, because original cell has 46. But we have to understand another important thing. If we take a cell, it has a one set of chromosomes, and then next one is a homologous chromosomes. So they exist in pairs. These are the homologous chromosomes. And in homologous chromosomes, they are not identical, but they are very similar. They are not identical, but they are very similar to each other. So what is exactly happening now? Let's assume like this. The blue color one is a duplicate of the first chromosome at the chromatid. And here the duplicated one of this one. So in reduction division, the meiosis, what is happening at the end, this chromosome is going to this cell. And this chromosome is going to these cells. Now you can understand the original cell has 46 chromosomes. And the 46 chromosome exist in pairs. So now 46 chromosomes we divide into 23 and 23. So this is get a one set, and this cell get another set. Right, now the blue color one is a duplicated version of this. So if we look number wise, it is 46, but the quality wise, the half of the chromosomes. So we call at this stage, it's diploid, and this is the haploid. Because originally there are 46 chromosomes with 23 pairs, and when it divided at the first stage, these pairs are separated and the homologous chromosome separated. One chromosome is going to one cell, other chromosome is going to one cell. That means genetic information, half is going to this cell and half is going to this cell. Because the blue color one with this arrow, it's exactly similar to the black color one. And they missed, this cell missed this chromosome, this cell missed this chromosome. The information, half of the information now divided. Then next stage is similar to the mitosis, then a prophase 2, metaphase 2, anaphase 2, and telophase 2. So these cells are undergoing next division, and at the end it makes two cells, and this cell also undergoing the division, and again it makes two cells. So once it divides, the one has this, and one chromosome is going here. So the chromosome number is 23. Chromosome number is 23 after the divisions. Right, I think you can start copying down when during this the starting. I think we can save some time. If you start to copying down rather waiting, I stop and then you start to copying down, it takes time. So you have enough time, right? So I will further summarize this event while you are copying, right? So we can divide basically meiosis into two stages. That is stage 1, prophase 1, metaphase 1, anaphase 1, telophase 1. And the second one, prophase 2, metaphase 2, anaphase 2, telophase 2. So reduction division, getting half of the chromosome actually happened at the first stage. Just to understand it, number wise, it seems to be not that useful to understand, because 92 after duplicated chromosomes, number is 92. And it becomes 46 and 46 at the end of the division. We don't see a reduction. And here we see reduction, 46 become 23. But actually we have to understand the numbers, not that numbers, but the quality wise, the homologous chromosomes, they are separated from each other. Because we learn from basic genetics, if there is a gene responsible for a certain character, for example, let's say capital A, and the homologous chromosomes, it has the other version of the genes, the alleles. Let's say here simple A. If they separate into two cells, this cell gets only capital A and this cell is get a simple A. Now you understand it's missing some of the genes. It's shuffled to this and shuffled to this. So that's why we call it's a reduction division, or one copy is coming to each cells. Then second division, it's exactly similar to mitosis, because it is the same thing happen what we learned in mitosis. All the events are same. After that, 4 cells are produced and each cell has 23 chromosomes. Right, now if we look at the significance of meiosis, these division take place in reproductive organs, the reproductive cells. So we call them gametes. Example, sperm and the ovum. So these cells finally represent the gametes, sperm and ovum. And one important thing is the 23 chromosomes. One cell has 23 chromosomes and the other cell has 23 chromosomes. So then sperm cell and the ovum, when it comes with the 23 chromosomes, they fuse to make the zygote. Then the zygote restore 46 chromosomes. So always when the offsprings are produced, the offspring has 46 chromosomes, children has 46 chromosomes, and 123 coming from father, 23 coming from mother, then they get the 46 chromosome. That's the one thing. Another important thing, that is the most important thing, we're going to look prophase 1 and metaphase 1. There are two important events take place, and the two important events is very important to form the variations. So we can understand even within a family, everybody is different to each other. The siblings, they are different to each other. So if we look this reduction division, okay, one chromosome is going to one cell, other chromosome is going to the other cells. Then always when it forms gametes, the homologous chromosomes separate. One chromosome is going to one side, other one is going to one side. But if this happen exactly the same way, there is no variation among the siblings. because fathers and mothers, the genetic information same, and always when they make the gametes, half is going to one cell, half is going to one cell. If this happen always in a fixed way, then there's no variations among siblings. So how we can really see the variation? If we understand this prophase 1 and metaphase 1 events, then we can get more clear idea about the variations. Okay, I can send this file as a single file if still you miss something. Right, now if you don't have any questions, we can move to the next event. Right, prophase 1 and metaphase 1. What other events take place to make the variations? Before I close this slide, just to summarize a few things happen in all stages. If you remember mitosis, at the end of the interphase, then the chromosome become condensed at the prophase 1. Then the chromosomes are visible, nuclear envelope disappear, nucleolus disappear. They're starting to disappear, and when it complete the prophase 1, end of the prophase 1, mostly nuclear envelope is not visible, nucleolus is not visible, and the centrioles start to form the spindles. Then when it comes to metaphase 1, all the chromosomes organize in the at the equator. Then anaphase 1, the chromosomes split from the centromere and pull to the opposite pole. And telophase 1, the chromosomes visibility decrease and they try to form the nucleus in at the both poles, and the nucleus, nucleolus and nuclear envelope start to visible. These are the major events happen at the mitosis. Now these things always happening in the prophase 1, metaphase 1, anaphase 1, telophase 1. And it is same here, prophase 1, metaphase, anaphase, telophase 2. At this stage is the same. So one hand it's easy, once you know the events at mitosis, that knowledge you can use here. So only the important thing now you have to remember at this stage, prophase 1 and metaphase 1. Right, let's see what is happening in prophase 1. This is the homologous chromosomes. Now what's going to happen, these homologous chromosomes getting very closer and closer, and then they almost at one time overlapped. So I can draw one overlap like this. So I make a different color of the next one. Here you can see it's overlapped. Now at this point, they are overlapped. So overlapping is very distinct at the end of the chromosomes, because they are very easy, the locations and the physical appearance, physical organization of the chromosomes allow to meet with each other at the ends. Because they are very flexible at the end, they can touch other chromosomes very closely at this end. Okay, now what is the importance of this overlap? part of the DNA molecules at the place it overlap exchange. So we can say like this: the blue color chromosomes, small piece of blue color chromosome is moving to the black color one, and the black color piece of chromosome is moving to the blue color one. So once it completed, we can visualize chromosome like this. So maybe here the black color one, part of the chromosomes is here. And maybe here another part of the chromosomes is here. And the other side, we can see the blue color one. And this stage is called crossing over. And also called a chiasma formation. The crossing over chiasma formation resulted the recombinant DNA. That means one DNA received another piece of DNA from another chromosomes. So the DNA is exchanged. That means some of the genes are shuffled. So let's say there are few genes located at this place, yellow color one, and the gene is moving to this side, and the genes which are on the black color chromosomes moving to the other side. So we can understand this is kind of a shuffling of the genes. So if it is happening in all 23 pairs, most of the genes are exchanging in random way. So it's exactly the shuffling of the genes. So this bring the variation. This is the first point, bring the variations. Okay, next one is the Metaphase 1. So here we see one event which produce the variation, exchange of the part of the chromosomes between homologous chromosomes, allow shuffling of the genes. Right, we're going to see the next one, that is a Metaphase 1. Right, in Metaphase 1, we have seen in meiosis chromosomes organized at the equator. This is also happening here, but the difference is the homologous chromosomes organized as pairs. Let's say the black color one represent one set of chromosomes, and the other one, homologous chromosome, is in front, like the blue color one. Maybe the other one, the blue color one is in the first side, and the black color one is in the next. The third one, still maybe the first one is a black color, a blue color, and the other one is a black color. Fourth one, the black color comes first and the blue color comes second. Now why is this? This is random. This organization is very random. And also we can understand, just give me 1 second. Okay, when a zygote is formed, 23 chromosomes are coming from mother and another 23 chromosome is coming from father. Then if we assume the black color one is a chromosome from father and the blue color one is chromosome from mother, so in this organization, if it is arranged very randomly, again this is chromosomes are shuffling. Because when the chromosomes moving at the end to this side and to this pole, there are different number of mother and father chromosome mixed up and they are moving to this side and moving to this side. This is also very random process and we call this random assortment. If this not happen, if the black color one always line up in this side and the blue color one line up this side, always one cell get completely father's chromosomes and the other cell get mother's chromosome. So there is no difference. So if it is arranged like this, mother and father chromosomes all are mixed in different way and it's a very random process. This is the second event, bring the variation. So these two events are most important to remember and it brings the variations in genetic variations to all the living organisms. As a result, one is not identical to the other one. So everybody has differences. The differences is important for the sustainability of the life. After that, what is going to happen same way, this chromosome is moving to this and this chromosome is moving to this in the anaphase. So then one pair is coming to one cell and the other pair is coming to the other cell, like here one. So two here. Now you see this resulted two cells. It has some number of mother's chromosome and father's chromosome. If you take all the 23 chromosomes, pair of chromosomes, then randomly they are allocated to these cells. Right, I think this is the most important points to remember in meiosis. And if you read the mitosis events, you can still apply that knowledge, the appearance of the cells during this first stage and second stage, they are same. In differences, it's a half number of chromosomes coming to one cells, and prophase 1 and metaphase 1, it brings the variations by random assortment and crossing over process. Okay, do you have any questions?